Introduction to recent advances in neurotechnology. Neuroscience represents a compelling area for modern research, and its goal is to establish understanding of the principles that underpin complex behaviors in animal species, including ourselves. For neuroscience methods, there are emerging techniques, for example, electrophysiology, pharmacology, optogenetics, optical biosensing, electrochemical biosensing. This figure implies that advanced engineering components serve as the foundational technologies for biocompatible platforms to support existing and emerging methods in neuroscience. They are not separate but have interrelationship. Among them, we are going to talk about frontier electrical, optical, and pharmacological technologies more deeply. First, electrophysiology. It is a branch of neuroscience that explores the electrical activity of living neurons and investigates the molecular and cellular processor that govern their signaling. Based on the location of the recording instruments, it can be divided in three types: extracellular recording, intracellular recording, and patch clamp. Electrophysiology has high spatiotemporal resolution about neural tissues from single cells and subcellular structure to large neuron populations at the level of tissues and organs. This figure below are the simplified physical model to determine the neural signal collected using an electrode. Neuropharmacology. It is the study of how drugs affect cellular function in the nervous system and the neural mechanisms through which they influence behavior. There are two main branches: behavioral and molecular. For complement to electrical forms of neuromodulation, pharmacological manipulation strategies can be an option in controlling neural behaviors based on endogenous receptors such as G-protein coupled receptors. Unlike electrical interfaces, these mechanisms support cell-specific operation. Approach for actively programmed and spatially precise drug delivery in support of this neuromodulation scheme include soft microfluidic networks, miniatured pumping system, and wireless control strategies. Combined with optoelectronics, it serves as the basis of enhanced options in spatiotemporal control when implanted with photoactivable drugs. For example, photoswitch is usually used. Then, combined with microfluidics, it increases its selectivity and spatial resolution. For example, there is DRADS. DRADS is abbreviation of designer receptors exclusively activated by designer drugs. DRADS allows GPCR to respond to synthetic ligands, not acetylcholine, which is their natural ligand, and is activated by clozapine and oxide. When we insert the virus vector into the mice or given an injection, the CNO works in a few weeks. For types of DRADs, we can see the two examples. The first one on the picture is an extensively used inhibitory DRAD. The second one is one of the earliest DRAD derived from the human muscarine receptor HM3.
Optogenetics is a tool to alter the neuronal activity. We need a genetic modification to do this, and the first step is to prepare the transgenic mouse. This transgenic mouse should express Cre companies under the same promoter of cell-specific protein genes. This ensures that cells expressing that protein will co-express the Cre companies. And then we inject the adeno-associated virus containing this gene. This gene has LOXP and opsin gene under a non-cell specific promoter. Wherever the virus is injected, the gene is transcribed. But the opsin is encoded in the opposite direction, so without fluxing the genes, it will transcribe non-functioning proteins. But some cells expressing the Cre companies will induce the LOXP ligation, which allows the opsin gene to be transcribed in a regular form. Then what is opsin? Opsin is a light-sensitive protein, and we mainly use these three opsins in optogenetics: tenorhodopsin, halorhodopsin, and bacteriorhodopsin. Tenorhodopsin is a cation channel which is activated in blue light and allows sodium influx. Halorhodopsin is a chloride pump activated in yellowish light and pumps the chloride ion inside. Green light stimulates the bacteriorhodopsin and pumps the protein out. CHR2 activation causes the depolarization, while the others induce the hyperpolarization. The genetically encoded fluorescent proteins are a powerful complementary tool to help us monitor the neuronal activity. During the signal transduction, the presynaptic neurons take up the calcium and secretes the neurotransmitter, and that neurotransmitter causes the postsynaptic neurons to fire. If we can monitor the sequence, we may visualize the synaptic activity. This type of protein sense the calcium ion and change their conformation, which makes them glow. And this type of proteins change their shape in a voltage-dependent manner. Some types of proteins have neurotransmitter binding sites, which let them emit fluorescence during the transmitter release. We need to distinguish between the fluorescent proteins and photoswitches. Fluorescent proteins are more like a responder of the signal transduction, while the photoswitches are the decision makers of signal transduction. With light stimulation, the activated opsins causes IPSP or EPSP, and this causes fluorescent proteins to light up or off. But in photoswitches, opsins are not involved in this process, and the switches directly recognize the light and change their morphology. These altered proteins decide whether the signal goes or stops. Every neuroscience method has their own strengths and weaknesses. These are the things that we need to consider. Electrophysiology has superior spatio-temporal resolution, while lacking the cell selectivity. On the other hand, the optogenetics have a high cell selectivity but having poor spatial resolution. But the emerging technologies enable the advanced technological components, which are groundstones of the neurotechnological platform and methods. The following videos will provide more explanation of how we overcome this type of limitations.